Hello viewers, my name is Mugai Gassan Mayo, and today I'm going to talk about cell injury. To begin with, the basic tenet of pathology. This is an idea that the pathologists before us had, that disease occurs due to cell injury, and this cell injury may manifest as functional changes or structural changes, and the functional changes may be observed as clinical signs, and the structural may be observed as lesions. Also, these functional changes can lead to structural changes, and functional changes can also lead to failure of vital functions or vital processes, which will further lead to cell injury. What are the causes of cell injury? First, we have hypoxia, which is reduced oxygen supply to a tissue. We have ischemia, which is reduced blood flow to the tissue. This can be caused when the blood vessel is blocked. Then it consequently leads to hypoxia and also reduced nutrient supply to the tissues. We also have physical agents. These are trauma, which is mechanical or heat. We also have radiation. Then the chemical agents are drugs, poisons, and insecticides or pesticides. Then infectious agents, these are basically living organisms. For example, the parasites, the viruses, the bacteria, the fungi, to mention them. Then we have immunological reactions. These are best seen in autoimmune diseases. We have genetic defects. Then nutritional imbalances. These can either be deficiencies or excesses. Then we have iatrogenic causes, which are basically the cell injuries caused by medical personnel. Then we have idiopathic, where we just don't know the cause of the cell injury. Then mechanisms of cell injury. We have first mitochondrial damage. This will lead to decreased ATP production, and it will lead to multiple reactions downstream. Then we also have an increased accumulation of reactive oxygen species. These damage proteins, DNA, and lipids, and other cellular structures. Then we also have entry of calcium. When calcium accumulates in the cytoplasm, it will increase mitochondrial permeability, causing the mitochondrion to release apoptotic enzymes. Then it will also lead to activation of multiple cellular enzymes, for example, the proteases, the ATPases that facilitate faster breakdown of ATP. Then we also have membrane damage. When the plasma membrane is damaged, we shall lose cellular components. Then also membranes like the lysosomal membrane may be damaged and bring about a release of the lytic enzymes that will digest the cellular components. Then we have protein misfolding and DNA damage. So when you have proteins misfolding, we may have median figures appearing in the cells, and also DNA. When DNA is damaged, we shall have reduced protein synthesis, and consequently we shall have activation of pro-apoptotic proteins. Let's look at the progress of cell injury. The cell's response to stressors or injury is not an all or nothing phenomenon. What this one means is that stressors or injuries, however mild, they can cause injury to the cells or adaptation. So however much the stressor or injury is mild, the cell will always respond to it. Then The response of a given cell to injury depends on the type, severity, and duration of the injury, and on the type and genetics of the cell. So, normally, a cell exists in a constant environment, and it's constantly managing the concentrations of fluids in its immediate environments. Then this is a condition we call homeostasis. Then when a normal cell is subjected to stress, it will undergo adaptation, and by this it will achieve a new steady state that is compatible with its new environment. If the cell cannot adapt, it will be injured. Then, also, when the normal cell is subjected to an injurious stimulus, it will also be injured. Now, this injury can be reversible if at all the injurious stimulus was mild or transient. Then the cell will undergo a reversible injury where it will go back to its normal state if the cause is removed. Then, if the injurious stimulus was severe and progressive, that type of injury caused will be an irreversible injury, and this will lead to cell death by two forms, either necrosis or apoptosis. So let us look at cell adaptation. So cells actively control the composition of the environment in and out, a state known as homeostasis. When cells are exposed to injury, they adapt to be viable in their new environment, hence acquiring a new steady state. And there are four basic types of adaptation: hyperplasia, which is an increase in the number of cells; hypertrophy, which is an increase in cell size; then atrophy, which is a decrease in the size of an organ due to a decrease in the number of cells or a decrease in the size of the cells, or both; then metaplasia, which is the replacement of one form of differentiated cells by another in the same tissue. So, manifestations of cell injury. We have hydropic change and fatty change. Hydropic change, some literatures also call it Claudius swelling, acute cell swelling, vacuolar degeneration, and some separate them, but always Claudius cell swelling and acute cell swelling will always be the same. But it is all to do with water influx into the cell, and it is the first sign of cell injury. It is caused by hypoxia, ischemia, toxins like cyanide. So, as you can see in the diagram, so this is ischemia, a blocked blood vessel. So that means we are having low oxygen supply to the tissues, specifically the mitochondria, which use up most of the oxygen. So that would mean that we have a decrease in oxidative phosphorylation, meaning also a decrease in ATP production, and that will lead to a decrease in activity of the sodium pumps, then which will lead to an influx of calcium ions, water and sodium ions, then an influx of potassium ions. So as these sodium ions are influxing into the cell, we shall have water following sodium. Then this will cause swelling of the cell and also the tubular organelles, and loss of microvilli and membrane bleeding. Also, ATP depletion leads to increased aerobic glycolysis, which will lead to depletion of glycogen stores and increased lactic acid production, which will lower the pH of the cell and consequently lead to clumping of nuclear chromatin. Then also ATP depletion will cause the detachment of ribosomes from the endoplasmic reticulum, causing a decrease in protein synthesis. Then we also have fatty change or steatosis. Sometimes it's called fatty degeneration. And this is basically the accumulation of triglycerides in parenchyma cells. Then this accumulation of triglycerides will occur due to increased free fatty acids inside the cells, increased synthesis of fatty acids, and decreased utilization of these fatty acids due to a resistance to insulin in conditions of diabetes and obesity. What do the organs that are affected by steatosis look like? First of all, it mostly affects the liver, heart, and kidney. Then, to identify these lipids, we need to avoid fat solvents, which are commonly used in hematoxin and eosin stainings. But you can use Sudan 3 or Sudan 4, which impart a red-orange coloration to the lipids. And this steatosis has forms, which are two. We have the macrovesicular and the microvesicular. The macrovesicular, as the name states, it means big vesicles, which means that they are large droplets of lipids and a displaced nucleus. So these large droplets of the lipids will push or displace the nucleus to the periphery of the cell. And this is the most common type of form of steatosis. Then microvesicular, as the name suggests, this means small vesicles. Then these are small and numerous droplets which can be seen, but they do not displace the nucleus. And this is much rare compared to macrovesicular. As I was saying in the slide before, this is macrovesicular and this is microvesicular. As you can see, these are large lipid droplets which pushed the nuclei of the cells to the periphery, as you can see in these cells. Then in microvesicular, the nucleus retains its position, but they are very many numerous and small lipid droplets. Now let us look at how this steatosis or fatty change develops. First of all, this diagram is showing the normal metabolism of fats or fatty acids. Then first we have free fatty acids coming into the liver cells. These are from the intestines or from the adipose tissue. From the intestines, they come in as chylomicrons from blood. Then that is step number 1. Then step number 2 shows biosynthesis of fatty acids. Of course, there are very many steps in here that are not shown. Then step number 3 is catabolism of these fatty acids, where they are converted to ketone bodies and carbon dioxide, and also to phospholipids and cholesterol esters. Then step number 4 is where we form now the triglycerides or the lipids. And step number 5, which is among one of the most important steps, is we are conjugating these lipids to the protein. And then step number 6, we are removing this lipoprotein from the liver cells and sending it into saturation to other tissues. But these steps all can be impeded, and when they are impeded or accelerated or magnified, we can have fatty change developing. For example, the entry of fatty acids a lot of free fatty acids get into the hepatocytes from the intestines as chylomicrons, most especially if you eat a heavily fatty meal. Then the step number 12, lipid biosynthesis. For example, if you have a condition of hyperglycemia, you have a lot of glucose in your blood, it will be brought to the liver so that that excess glucose can be converted to lipids and stored. Then the catabolism, if enzymes that are responsible for converting these fatty acids to ketone bodies or to phospholipids or to cholesterol esters are destroyed or denatured, these processes can be cut off, which will lead to an accumulation of fatty acids, and consequently these fatty acids will follow step number 4. And step number 4 is the esterification reaction, where we have fatty acids and alpha glycerophosphate combining to form the lipids or the triacylglycerols. Then this number 4 will occur in conditions where we have a lot of glycerol phosphate. Then number 5 is the conjugation. If you have decreased concentration of apoprotein, maybe it is not synthesized, you will impede this reaction where the lipids are converted to lipoproteins, and so you will have an accumulation of the lipids in the hepatocytes. Then also, if the hepatocytes fail to release the lipoproteins due to maybe membrane damage, you can have these lipoproteins converted back to lipids, and hence you will have lipid droplets filling up in the cell. So now, reversible injury. As earlier said, reversible injury can be caused by hypoxia, but once this injurious stimulus is removed, the cell will revert to its normal state. So you have your stressor here, and it will cause cell swelling and swelling of the tubular organelles, also leading to chromatin clumping as we saw earlier. But if normal oxygen levels are restored in the tissues, then the cells will revert back to their normal state. Now, irreversible cell injury. Basically, what you need to know about irreversible cell injury is that the cell has reached a point of no return where it will only progress to death, which can either be by necrosis or apoptosis. So however much the injurious stimulus is removed, it will not go back to its normal state. Now, this diagram is showing an example of irreversible cell injury, how it may progress. So you have a condition of persistent ischemia. Then it will decrease oxidative phosphorylation, thereby decreasing ATP production, and this will increase calcium influx in the cell. And once you increase calcium in the cell, once calcium accumulates in the cell, this calcium will activate phospholipase, which will lead to cell membrane damage and mitochondrial damage. And it will also lead to formation of Melian figures. We also have calcium activating proteases, which will damage the cytoskeleton, leading to cell membrane injury. And we have endonucleases. Endonucleases are enzymes that cleave or digest DNA. So we shall have those nuclear changes: pycnosis, karyolexis, and karyolysis. Then we also have the lysosomal hydrolytic enzyme leakage, which will cause lysosomal membrane damage and leading to autolysis and cell necrosis. Thank you for watching. My name is Mugaiga Asenonamayo, and feel free to tune in again to our YouTube channel, as we shall keep on posting more videos to help ease your learning process. Thank you.